The approach that we're going to be talking today about has been derived to compare properties and functional activities of tissue-derived and peripheral blood T cells. The significance of the topic is exemplified by the fact that peripheral blood T cells represent only 20% of the total T cells in a body. Being able to study a small number of tissue-derived T cells is very essential, and that's exactly what we have achieved.
The main advantage of this technique is that it requires smaller number of cells and smaller volumes of reagents than traditional techniques.
Begin by using polypropylene scissor-type forceps to submerge glass cover slips in freshly prepared acidic piranha solution for 25 to 30 minutes. At the end of the wash, rinse the cover slips 7 times in a fresh volume of ultrapure water for each wash, and set the wet cover slips aside to let the remaining water roll off the clean glass. When the cover slips are dry, aliquot 2 microliters of the final liposome mixture precisely in the center of a self-adhesive slide channel within a sterile flow hood, and immediately but carefully align one clean and dry cover slip with the slide to allow the cover slip to be gently lowered onto the sticky side of the slide, and use the outer ring of the polypropylene scissor-type forceps to apply a gentle pressure to the peripheral contact of the cover slip with the slide, making sure that the slip is tightly attached to the slide to prevent